Hi, my name is Eddie Wild and I am a technician at Kansas State University in Dr. Keith Gido's lab, and I will be presenting our research over the behavioral response of Loach Minnow to non-native predators. Before I get started, I'd like to thank a few people. I'd like to thank Bill Stewart from the Bureau of Reclamation for funding the project, Josh Walters and Sky Hedden from Arizona Game and Fish for providing the Loach Minnow and helping out with transportation, the Konza Prairie Biological Station for housing the Mesocosms, which is where this experiment took place, Mary Richardson and Vanessa Berg for helping out with the permits, and Tom Turner, Alex Cameron, and Dave Probst for their discussions on the experimental design. Loach minnow populations are on the decline. As most of you probably know, Loach minnow are an endangered species, and I just wanted to give a quick visual representation of that. So the map on the left represents collections from 1980 to 1999 in the Gila River drainage, and the green dots represent abundance and the red X's represent absence. And then in the same format, the map on the right represents collections from 2000 to 2007. And from these two maps, you can see that there is a clear decline of Loach minnow populations, but the mechanisms attributed to this decline are often questioned. One of the main mechanisms that is often attributed to Loach minnow decline is the introduction of non-native predators, and Crosby Hedden from the Gido lab previously displayed this by showing that when bass are not present within reaches where Loach minnow and spiked dace are present, Loach minnow and spiked dace are much more abundant where bass are not present in comparison to where bass are present. So there is that clear relationship. Loach minnow are a riffle specialist, and they likely co-evolved with two main predators, the round-tailed chub and pike minnow, which are mainly pool and run specialists. And so with that, non-natives offer an additional suite of predators, which begs the question, how do Loach minnow behave in response to non-native predators? So that's our first question. Is there a behavioral response of Loach minnow to the introduction of a non-native predator? And does the response differ between smallmouth bass and flathead catfish? We'd also like to know, is there a difference in the movement or consumption rate of the two non-native predators? So we did that by collecting Loach minnow from the ARCC in Arizona, and those Loach minnow were then transported back to the Konza Prairie Biological Station in Kansas, and smallmouth bass and flathead catfish were collected locally in Kansas. And because Loach minnow are an endangered species, I feel like I have to say that the fish were genetically crossed from lineages of two separate populations, so they could not be released into the wild, unfortunately. For our study design, we used 8mm pit tags, and when you combine that with submersible antennas placed in mesocosms, you get a fine-scale behavioral study. I'm sure most of you are familiar with submersible antennas, but if you're not, with a 8mm pit tag in a fish, we can tell exactly where a fish is during a given time when that fish swims over those antennas. To give you a bird's eye view of that design of the mesocosms, this is kind of what it looked like. So we had 3 pools and 3 riffles in each experiment, and a trolling motor with a flow tube which pushed the flow through the entire mesocosm. And then we placed submersible antennas in each pool, so 3 submersible antennas in the experiment, and there were no antennas in the riffles. So if a fish was not being detected, then we assumed that it was in the riffle. We assume that because if it's not being detected, then it's likely not in the pool, and so it must be in the riffle. We had 10 individuals in each experiment with an equal sex ratio. So 5 males, 5 females, and unfortunately we were only able to do 5 replicates because we did not want to use individuals more than once. So we had 2 replicates with the introduction of a smallmouth bass and 3 replicates with the introduction of a flathead catfish. We had a 48-hour control period where no predator was present, we just observed baseline movement. And then following that 48-hour period, we introduced either a flathead catfish or a smallmouth bass with its mouth zip-tied shut. And we did that just to control for consumption. Then following that 48-hour period, we had 48 hours where the predator was able to eat and we snipped the zip tie. For our statistical analysis, we used R software, but due to the low sample size, we will just present the patterns of the observed data today. So one of the limits of this study was that there was a lot of individual variation. There was variation in movement across treatments and trials in loach minnow and predators. And to show you what that looked like, I made this graph where the y-axis represents upstream to downstream movement, and the x-axis represents the time series of which the experiment took place. So loach minnow is represented by the red and flathead catfish is represented by the blue line. And this is one individual loach minnow and one individual flathead catfish movement. So we can see that before the flathead catfish is introduced, there is much more loach minnow movement than after the flathead catfish is introduced for this individual. So initially, a load of movement during that 48-hour period, and then the loach minnow just kind of stays in that top pool, the most downstream pool. I'll let you watch that one more time. However, for this individual's movement was much less before the flathead catfish was introduced, and then after the flathead was introduced, there was much more movement. And so that just gives you a good idea of the individual variation in movement. Another takeaway from this graph is that flathead catfish and smallmouth bass tended to move most often at night. And to give you an idea of time, each grey bar represents midnight of a different day. And so you can see that the flathead catfish tended to move most often during the night. So for our first question, do loach minnow respond to the introduction of a non-native predator? We answered that by looking at if loach minnow select pools without predators. And we did that by developing a selection index. And to calculate that selection index, for each 10-minute time period, we took the number of fish in the pool with the bass, and we subtracted that from the number of fish in any other pool. And so, for example, a selection index value of 0 would mean that the maximum number of fish in any pool is equal to the number of fish in the pool with the bass. A selection index value of -1 would mean that the number of fish in the pool with the bass is 1 less than the number of fish in a different pool. And so on. So a selection index value of -2 would mean that the number of fish in the pool with the bass is 2 less than the number of fish in a different pool. And because the next graph is pretty messy, I wanted to give you an idea of what we really expected. So we expected that loach minnow would select pools where bass were not present. And so that would mean that we would get more negative values than values that were closer to 0. So a value of -4 would mean that there are 4 less fish in the pool with the bass than there are in a different pool. However, this is what our results looks like. So again, the question is, do loach minnow select pools without predators? And just to run through this graph quickly with you, this is a histogram, just a pretty simple histogram. So along the y-axis is frequency, along the x-axis is the selection index value, and then flathead catfish and smallmouth bass along the top, and then the two treatments of a predator present with a zip tie in its mouth on top, and a predator consuming with the zip tie clipped along the bottom. From this graph, there's two main takeaways. The first is that Loach Minnow did not avoid pools with smallmouth bass, and the second is that Loach Minnow may have taken refuge in the riffles during the flathead catfish trials. You can see in that top right that Loach Minnow did not avoid pools with bass because most of the values in the smallmouth bass trials were skewed towards 0. However, in the flathead catfish trials, Loach Minnow may have taken refuge in the riffles, and we just assume that because there's far fewer detections. And if there's far fewer detections, then that means that the Loach Minnow were likely not in the pools because they weren't being detected, and if they're not in the pools, then they're likely in the riffles. Another way we answered the initial question, how do Loach Minnow respond to the introduction of non-native predators? We used movement as a variable, and we expected during the control period for movement to be highest, and then when we would introduce a predator, we expected movement to reduce greatly. And just to run through this graph with you real quick, we have the two treatments along the side. So the control is on top, no predator present, a predator present in the middle with a zip tie in its mouth, and then a predator with the ability to consume on the bottom. And again, same thing, smallmouth bass are represented by the green and white, and flathead catfish are represented by the three other colours. And then the y-axis represents number of individuals, and the x-axis represents number of movements. So is there a behavioural response of Loach Minnow to the introduction of a non-native predator? Movement did reduce during the introduction of a bass, but did not change with the introduction of a flathead catfish. We reason that this may be due to individual variation during the control treatment. And what we mean by that is that during the control, all variables were the same, and so we expected the control movement in the flathead catfish trial to be similar to the control movement during the smallmouth bass trial. However, they really weren't very similar at all. And so individual variation is obvious during the control trial. For our consumption rates, 4 of the 50 Loach Minnow were consumed over 480 hours of predator presence, and 3 were consumed by a flathead catfish during the 3 flathead catfish trials, and 2 were consumed by, 1 was consumed by a smallmouth bass during the 2 smallmouth bass trials. Smallmouth bass moved an average of 45 times per trial, and flathead catfish moved an average of 29 times per trial. So there were a few limitations to this study. One of the limitations was that these were hatchery fish, and hatchery Loach Minnow may behave differently to wild Loach Minnow. The second limitation, which I've already discussed a lot, is just the sample size. And there was a lot of individual and replicate variation, which is especially problematic within the control groups. So to conclude, much further research is needed. We do need to increase our sample size. Loach Minnow movement responded more to the introduction of smallmouth bass. However, Loach Minnow selected pools where smallmouth bass were present. And these two results don't really coincide. They don't really make much sense. If Loach Minnow movement responded more to the introduction of a smallmouth bass, then we would expect that Loach Minnow selected pools where smallmouth bass were not present. However, that's not what we found. And that's likely due to the individual variation. And then flathead catfish may be a more efficient predator than smallmouth bass of Loach Minnow. However, that's difficult to say with such a small sample size and so few trials. So these results suggest that Loach Minnow may take refuge in riffles when predators are present. We just conclude that just because there were far fewer detections during the flathead catfish trials, and that may be due to the Loach Minnow being within the riffles. So for our future intentions, we intend to increase our sample size. And we'd also like to compare the movement of Loach Minnow to other riffle specialists, like those within Ethiostoma. I look forward to any questions that you may have, and thank you for your time.